This is a continuation of Biochem Lecture 2 of Exam Block 3, covering more chromosome abnormalities. Made some more pictures to kind of help remember some things and explain some other things. So first of all, this part we're talking about at first here is duplication syndromes. So example of the first one we're talking about is called Charcot-Marie-Tooth Disease. I'm guessing these are 3 people that were involved in it. So what did I do here? I drew Marie with 1 tooth. Okay, so Marie-Tooth disease, this is Marie with 1 tooth. This only happens about 1 in 2,500 people. And with this one, it's a duplication of part of chromosome 17. So I drew here Marie with her 1 tooth is holding a 17 magazine in her hand. And she has weakened arms, because apparently one of the common symptoms of this is, I'll just read up the things that they put down. Duplication of the small arm of chromosome 17, most common inherited disorder of peripheral nervous system. Peripheral limb weakness, high foot arch, hammer toes, 1 in 2,500 live births, due to defective PMP22. PMP22 interacts with gap junction connexins to regulate nutrient transport. Overexpression of PMP22 closes connexon and promotes demyelination. So I drew here PMP22, which is a pump with a tutu on it. And I have her with these high arches and hammer toes, really weak arms. I don't know, I'm trying to think of some way to remember that it was a disorder of peripheral nervous system, so I drew her hands on fire. That's kind of a dumb way to do it. So the pump is overexpressing, so it's blowing lots and lots of air. And yeah, hopefully that helps remember the stuff. Okay, so this next one. This is about Mech P2 duplications. So here we have this mech robot guy. Those are P's, I guess, on his head. This typically happens in males, and it's the long arm of the X chromosome. So here this is Mech P2, and he's got, put a 2 on his head too, Mech P2, and he's got the X thing in the middle and the long arm reaching out, and it's grabbing the methylated DNA. So for this one, they have difficulty standing or walking, so he has these like robot wheels for feet, which would make him hard to sit down or to walk, I guess. I'll read what he has on this one as well. Usually in males, moderate to severe mental retardation, problems walking or sitting or feeding, duplication of material on long arm of X chromosome. Mech P2 genes codes for protein that binds methylated DNA. So there he's grabbing this methylated DNA. Okay, so the next part here is going to be, we're going to talk a little bit about sex determination disorders. And so it looks like, from what I can tell, is that around this, or what he says, it sounds like the 6th week of development, the baby's gender is identical. It's a male or female. The only way you could tell it apart is if you did like genetic testing, because physically they look the same at 6 weeks. And then this SRY gene comes out. So sorry is what it was like to me. So sorry, I have to tell you, you're going to have a male. So if SRY comes out, it's going to be a male. So what SRY does is it triggers this TDF. TDF stands for testes determining factor. So SRY activates the testes determining factor, and apparently there's like the testes determining factor, and then there's this DAX1, and the testes determining factor gets stimulated a lot. It overcomes this DAX1, and that's going to determine that the testes determining factor is strong, whatever, and that's going to activate SOX9. I don't know exactly remember that. Maybe think sex 9. That looks like, I don't know, SOX9. and that's going to activate the Sertoli cells, and the Sertoli cells are going to start to release. You read it on here. The Sertoli cell, the TDF upregulates SOX9, stimulating Sertoli cells to produce ABR, or androgen binding receptor, as well as MIF, Mullerian inhibitory factor. So Mullerian inhibitory factor and androgen binding receptor. Also, this testosterone factor stimulates the Leydig cells to release androgens, which help with the physical male sex characteristics like the penis develop, and also AZF, which has to do with spermatogenesis. So if there's problems in this, it's going to probably be with the issues with SRY gene. For example, if there's a deletion, there's no SRY gene, even though they may be, an XY, if there's a deletion there, then they're going to have not normal sexual characteristics. That's actually more expressed a little better on the next slide here, so let me turn to that. So it says here with if there's an XX female with a male phenotype, in other words, if you have somebody that's genetically an XX, but they look like a man, then what's going on here is that the SRY gene was probably translocated to an X chromosome. So what you have here is an XX person that somehow got that SRY gene. If they have the SRY gene, then they're going to have testes termination factor, whatever that was, an SOX9 gene, the Sertoli cells activated, or they're going to basically turn into a man, even though they have XX gene. And they're going to have small testes and be sterile, I guess. Apparently there's something else called De La Chapelle syndrome, which is that thing that I just described. That's what they call it, De La Chapelle. Okay, next also the XY. Sometimes you can have an XY who has a female phenotype. In other words, this is genetically an XY male that looks like a female. So how does that happen? Well, like I was explaining earlier, if the man loses this SRY gene, he's not going to develop into a male, so he'll look like a female. Or if the SOX9 gene that's supposed to stimulate the Sertoli cells to release the MIF and the ABR, then they're not going to develop those primary male sex characteristics, phenotype, whatever. So, is that everything on that one? I can't remember. Yeah, and there's something else called Swire Syndrome, SWYER, and that's where somebody essentially is an XY, but they have gonadal dysgenesis, so it makes them sterile. Okay. Next, what we have here is a little bit about pseudo-hermaphroditism. So it's a pseudo-hermaphrodite. So in this case, we're going to talk about the female one first. This is somebody who has 46 chromosomes, XX chromosomes, but in this condition, they only have the gonadal tissue of one sex that matches the karyotype. So that what's going to happen with this is apparently this constantly happens due to congenital adrenal hyperplasia. So sounds like overgrowing of the stuff that makes I shouldn't guess on what's happening here. I don't know. Congenital adrenal hyperplasia. So hyperplasia means overgrowing adrenal. I think that usually has to do with, I guess it could be adrenal glands, but also maybe it has to do with androgens. I don't know. So while they're developing, that overgrows. Also, there's something that associated with this called salt wasting syndrome, whereas basically they're producing too much aldosterone. Actually makes sense. The adrenal glands overgrowing and that overproducing aldosterone. Okay, this is starting to make sense. So salt wasting syndrome is from that. So let me read what it says on here. Clitoral enlargement, labial fusion to form scrotum-like structure, salt wasting syndrome, aldosterone lack. Oh, so they don't have enough aldosterone. Okay. So that, Duh, of course, it's all wasting. We don't have enough. Okay, so they don't have enough of that. And it's due to deficiency in 21 hydroxylase needed for normal glucocorticoid and mineralocorticoid biosynthetic pathway. So they don't have the 21 hydroxylase. How can you remember that? Think of a 21-year-old female who really wants salty things. I don't know. Maybe that would help. Okay, the next one here is male pseudohermaphroditism. And the same thing again, 46 chromosomes, but this in case it's in on XY, but they appear, they're a pseudohermaphrodite. So again, it's a gonadal tissue of one sex that matches the karyotype, and normal testicular development, but completely masculine or female external genitalia, or incomplete, normal testicular development. So they have normal testes, but incompletely masculine or female external genitalia. So basically, it's a man who has a vagina or maybe just a little tiny penis or something like that. And it's usually deficiencies in testosterone. So remember over here, the SOX9, the Sertoli, stimulates the Leydig and all that. Anyway, so these steps down here is what kind of stimulates the person. I think it's actually this one that stimulates the testosterone, and the testosterone is what stimulates the growth of the penis. And so if the person isn't expressing enough testosterone, or their cells seem insensitive to the testosterone for some reason, then they're going to have an underdeveloped penis, or male sex characteristics primary. And also this has to do with due to deficiencies in testosterone synthesis, metabolism, or androgen receptors, 5-alpha reductase deficiency, 5-ARD. So they have small penis, blind vaginal pouch, or testicular cancer. And then the other one here is androgen insensitivity syndrome, defective receptors or lack of receptors. This is 1 in 20,000, XLRAIS. I don't know what that stands for, and this is testes within the abdomen. So in this case, rather than the testes descending down here, so they start out up here, and this is where, of course, the ovaries are for women. Well, it's more like right here. And so they're supposed to descend down and go into the testicular sac, but they don't. They stay up here, and that's causes the problems. And that's what I got.